On to the whole sensors. So these are cells that can be tailored to respond to different compounds. So these are different from whatever synthetic biology work I was showing you earlier. And then you can see this chemistry is very simple, as most studied some cell biology or biochemistry, where we have studied lactoperon, that if lactose is present, beta-galactoside enzyme is made. So similar concepts we employ, but this concept we find these promoters or genetic regulatory elements for each and every target that we want to do. The purpose is, if the repressor is there and the target comes in, it throws the repressor and the gene expression can go on. This gene expression now can be scored as either as a fluorescence or as a luminescence or as an amperometric change also. So this is what our group has now expertised in the past 5 to 6 years, and some results I'm going to share with you now. So the primary thing that we do in whole cell biosensor is we need a lot of colors, because if we are doing multiplexing or trying to detect more than one kind of bacteria or targets, then one color won't be sufficient. So for our rescue, the Nobel laureate Robert Sien actually developed these different fluorescent proteins, and we use few of them to do multiplex detection of mixed targets also. So the proteins that we use right now are M-cherry, GFP, RFP, and yellow fluorescent proteins. And we use a mix of chemical genetics and also bacteria. So anytime any target is going inside the bacterium, say for example if it is an antibiotic, bacteria try to resist it. So what they do is they throw it out. They are known as pumps, expeller pumps. So we also use chemical genetics and knockout to make sure that the bacteria is not throwing the target out and the target is available for turning the gene expression on. So let's see some of the work, and then there are certain features that are required for whole cell biosensors, and when we work on them we make sure that they are sensitive, they can detect very low out of the target also, specific. So if there is an biosensor for a particular amino acid, other 7 to 18 or 19 related amino acids or other amino acids should not be able to interfere with the results. It should have a good dynamic range and they should be stable also. These are some of the positives of whole cell biosensors that we find, and we try to stick to these. So not the whole of the picture is also not rosy. I am going to go through the demerits only. The toxicity. If a particular compound is highly toxic for the bacterial cell, we cannot turn it into a whole cell biosensor. Then if the regulatory elements are not available already, then it's difficult to make a biosensor. But I will show you some results where we have overcome this problem also. Now, so there are several applications of these kind of biosensors in environment, in diagnostics, even in biology. Small molecules, small metabolites can be now monitored using biosensors. Okay, so going on to now synthetic biology part, because this is where we got high success. So there are now parts available, like ribozyme bonding sites are available. There are different types of reporter proteins available, and there are some genetic elements also available we can use for our work. So transducer optimization, genetic element optimization, protein engineering or evolution can also be done for generating these kind of biosensors. Now, so this is what I was just telling you earlier, and let me go on to the next part where we made an biosensor for detection of a fluoroquinolone antibiotic. So for this, we used a small peptide which is toxic. So this toxic peptide, which is known as IBSC, actually if it is expressed in E. coli, the cell dies. What we have done is that part, we have cloned a library of random sequences here so that the IBSC protein is not expressed. Now if this is a genetic element and if it is getting induced by a particular target, the cell will die, otherwise the cell will be living. So this is what is shown over here. We did a mixed low throughput kind of an experiment where we cloned 10,000 fragments in this vector, and then we screened and we found one which was getting induced and the cell was dying because of expression of this toxic peptide. So the story was followed further and we had the knockouts. We wanted to know what is the mechanism of this gene control by this peptide, and we also showed that okay, you take a lot of knockouts and then you transform this plasmid, the previous plasmid, in those knockout of E. coli, and then show that yes, we found that one protein is there, HNS, which actually if it is not present, the regulation is gone. So we did some assays also, we purified this protein, did assays with different variants of that regulatory element to show that this is the element which is actually controlling, really controlling the gene expression. So by doing more experiments or mutations in these 10, -10 and -35 regulatory region of that element, synthetic promoter, we find anything you do here or here, the regulation is gone, regulation gets messed up. So this is the mechanism we proposed in this nucleic acid research paper we published, that in the presence of, if fluoroquinolone drug is not there, this HNS binds to this regulatory elements and keeps it silent. The moment fluoroquinolone comes, this is kicked off and the expression of this peptide goes on, and that is where we turn this into a very sensitive biosensor for fluoroquinolone category of drugs. So this is what it is. The whole scheme is shown over here. And as you can see, I was telling you that these biosensors have to be very specific. They should not be turned on by any other category of drug. So this is the data which is shown over here, that only fluoroquinolone category of drugs are able to induce this protein. So this is again that mechanism which is explained over here, that HNS protein usually binds and keeps it like this, but fluoroquinolone kicks it off and the gene expression turns on. So this is one of the way by which we can use synthetic biology approach for making biosensors for antibiotics. So my group has also worked on these are summaries of individual papers that we have been able to publish, and so this was a paper that shows we made a biosensor for SDS. SDS again, it has a GFP. This is the regulator for that, and when the SDS is present, the cells are like this, absent cells are like this, and when SDS is there, the GFP expression goes on, and you can see in absence of SDS, there is no fluorescence in the bacterial cells, and in the presence of SDS, the cells show fluorescence. So these experiments were then followed by fluorescent assorted cell sorting experiments also, and we showed that SDS only can induce this gene, and otherwise this biosensor stays silent. So again, it is showing that these are very sensitive whole cell biosensors. So a lot of people these days are also working on plant metabolites, and plant metabolites are excellent antibacterial agents also. They are antioxidants also. So we wanted to assess some of the mechanism of action and the biosensing potential of some of the regulatory elements that one of my students, Rajat Dhyani, who is in NIH right now doing a postdoc, did. So Rajat actually made a heat map of this, and he tried several biosensors. He made a battery of around 15 regulatory elements, and then he found out that 3 of them are being induced by different metabolites. And this is the success story of that biosensor. So we have used this chemical genetic approach to assess the secondary metabolites. So we have used only few of them, but it is now easy to think that any plant metabolite we could be targeting this way. We can assess the purity. We can also assess the mode of action of that plant metabolite in terms of how it affects the bacterial cell. So this is now going into the health side, homogenetic acid based whole cell biosensor we have developed, and this actually detects the L-captonuria disease. L-captonuria disease, homogenetic acid is made in that, and the high amount of homogenetic acid is there. So this the same way we isolated one genetic regulatory element, and then this is our reporter protein, and you can see if homogenetic urea is present, it is showing good fluorescence, and if it is not present, homogenetic is not present, then it is there. We also assessed these on some of the doped samples. We made synthetic L-captonuria disease samples in urine, because it is secreted, homogenetic acid is secreted in the urine, and we could detect the disease fairly accurately. So this is again 2 more diseases, and I'm presenting this because again in physiology a lot of errors, metabolic errors are there. So these kind of biosensors are very well adopted to be used for quick screening of diseases in case of livestock animals also. So where cystinuria and urea could be detected using these battery of biosensors, and using a fluorescent plate reader it is easy, and within 30 minutes to 1 hour, depending on what is the fold change in fluorescence in particular biosensor, we could detect it. And this work also made to this reputed biosensor and electronics journal. So this is another work that we have taken now along with the biosensing of particular metabolites, xenobiotics, antibiotics. One of my students, he has actually made a degradation system also. So degradation system is a separate story. I'm going to talk some other time. But what I am telling you over here is that we have made a ppb level, a parts per billion level of antibiotics biosensor based on whole cell biosensing. And now that Shubham had developed a degradation system based on some nanoparticles, actually now we can monitor degradation of antibiotics in the environment also using these biosensors. You can not only now monitor the presence of antibiotics, if you develop a degradation system, you can follow the degradation profile also, kinetics also, using these biosensors. So again, the point to be noted here is that the sensitivity is super high. So anything we want to monitor in case of environment, these kind of biosensors could be of good use. So this is the last work that we have done. We have sent it for review as of now. So this is for histamine detection. Histamine is expressed a lot by bacterial contamination. It is converted. This toxin is the most important toxin in seafood actually as of now. So my student Ankita, she has made this biosensor where she showed that highly sensitive and specific histamine biosensor can be made. And then as you can see again over here, when the histamine is absent, there is no fluorescence in the bacterial cell, and if the histamine is there beyond a limit, then these cells light up. And you can see histamine is an analog of some of the amino acids. You can see any amino acid you add, there is no fluorescence. But when histamine is there, this shows. So this is where the biologists need to be proving their point because they are highly specific, sensitive. Now what we are doing as of now is we are not happy with the time that these biosensors are taking to show the signal. So it's like varying from anywhere from 30 minutes to 2 hours. We are adding now some amplifying modules, again recombinant amplifying modules, by which we hope that we should be able to do the detection within 15 minutes. So the next frontier that my group is undertaking is targeting some of the cancer metabolites, and then similar battery of biosensors we are trying to make so that we can detect cancer early, reliably. So the purpose is not to replace the normal cancer tests. Actually, the purpose is to complement them. When there are 5,000 samples to be tested for a particular kind of cancer, we can reduce that number to 100 or 200 using these kind of biosensors. That would be a great help to the society and less financial burden on people. So with that, these are some of the futuristic ideas that I have, but due to lack of time, I am going to skip it, and kindly I apologize in advance, but you can read it. This presentation will also be here later on if somebody is interested. And I am very lucky and very thankful to funding agencies who have supported us in the past 13-14 years since I joined IIT Roorkee. ICR especially I thank, because most of my money has come from NAIP, ICR, and right now NASF funding my research. DBT, MHRD, ICMR has recently funded a project on millets, and so thank you so much for your attention. And if there is any quick question, I will be more than happy to.
You have a very excellent work and presentation as well. Only my query is that is there any commercialization of any of the products that the biosensors or any that is the key develop, particularly detection of urea in milk, which has the practical implication.
Right. So urea biosensor right now is being evaluated by GSK. Okay. So there are certain issues. They have come back to us. Now we are trying to sort those out. So that's one. So about other, we did not find any commercial part so far, but we are ourselves, as told in the last couple of slides, we want to make them more sensitive. So sensitivity is still an issue before I go to companies and tell them, okay, this is an excellent product. So if So if I can bring it down to at least half an hour, it will be in this. And at the same time, we are also trying to optimize how we collect these cells. So we are using, I'm going to buy one encapsulator in coming month. We are planning to put these cells, these recombinant cells, encapsulates alginate beads, and show that this, in the lab scale it has worked, but I want to make it uniform so that batch to batch variation in the biosensor is not there.
For example, urea in the milk.
So using aptamer, urea and milk within 5 minutes.
How much cost maybe?
So yeah, so actually NASF asked me to do the costing at the time. So if you take the cost of development aptamer, because once it is made, that's done. Government has already given money. So once you have done that, then probably 4 to 5 rupees per test. Yeah. We don't want it to be 40 rupees because milk is only 50 rupees. So nobody will be, yeah. So we have to bring it as well.
What's about accuracy and repeatability?
Accuracy of?
Repeatability.
Okay, so I think made 4 points. Accuracy in terms of interference from related molecules. So for urea, I can tell we used thiourea, it does not detect. For the amino acid, I have shown the data. For histamine, only histamine. You use any other related molecule, it doesn't, because it is working on the principle of genetic regulation. So they are highly specific, transcription factors are highly specific. So does it answer your question? Yeah? Sorry? Glucose detection. But we have not, see, for glucose detection, already cheap methods are available. What I can tell you, one group from Middle East, they used our urea aptamer and they used it to detect urea in urine and publish detection. So, and again,
my point was you have touched a niche. Those are the niche diseases that kept in urea or the homogen diseases. Those are not very common problem, I guess.
definitely, yeah, sure.
That is much more common where you can go for better standardization and more verification of this of the new technology. Isn't that the case?
Yeah, but again, you see, so there are two things.
Yes.
One is I am targeting something which is, to be used by people. Another is,
Certainly demonstrating a technology is one aspect of moving into the public.
right.
And
So it will be easy. It won't be any difficult. Actually, glucose oxidase based sensors are already there, right? And they're cheap also.
Yes.
So you have sticks. So I mean, why do I have to invest my time and resources in doing that? I mean, this was my logic. Okay. But we could target something else in diabetes for sure.
You can
HB1AC like, right?
We can track hormone level.
Sure, we can do that. We can do that. That is possible for sure. Good.
So, if there are no questions, then I thank Professor Naveen Kumar for a nice presentation on biosensor technology. Now I request the organizers to felicitate this lead speaker because he has to leave early immediately.
We request you, sir, Dr. Rastogi, to please felicitate him with a moment. Powerful chair again.
So next speaker in this session is Dr. P.S. Yadav. I request him to start his presentation.
Good afternoon, everybody. I am thankful to the organizers for giving this opportunity to share our research with our SAPI colleagues. I will be presenting the developments in animal cloning, mainly in buffalo, because in India, buffalo is the only animal which has been cloned till now. So you are listening in the morning also that in 1997 first clone was produced Dolly, and in the subsequent years India also tried this technology on our own animal buffalo, and first clone calf was produced in 2009. First thing was to see the feasibility and possibility. Then question came that how we can make use of technology, and what research have been conducted in India, where we have reached, I will share. Animal cloning is an asexual means of reproduction. It is the genetic copy of a donor animal. It is similar to identical twins, and it has a predetermined sex because the donor cell decides the sex of the clone animal. Normally, without micromediator, the cloning is done. There is not much basic difference. The basic concept is same. We have to remove the nucleus of the mature oocyte either by piercing the zona pellucida and sucking it out, or cutting the nucleus part so that we can have the mature cytoplasm, which have the potential to reprogram the donor cell back to the germ cells. So the method which we are using here, we collect the somatic cells from the animal. These days we are collecting from superior males or females. Then cells are grown and cultured, and one of the somatic cells is taken as a donor cell. From the other side, we take the ovary, we collect the immature oocytes. From the immature oocytes, we mature them, then make them zona free. Then after making zona free, there is a bulging on one side. So we can cut it with a blade so that we can have the cytoplasm which is without nucleus. So two such cytoplasms or even one cytoplasm can also be fused with the donor cell, and then it is activated with chemicals, and this way the process of development of 2-cell, 4-cell embryo continues and it reaches up to the blastocyst. At blastocyst stage, it is transferred in the animal as in case of other embryo transfer or IVF embryo. After 30 days, we check the pregnancy with ultrasonography and also continue to check it 2-3 times to ensure that it is pregnant. And if the calf is born, then we go for testing that it is belongs to the donor cell or not. Some facts about the cloning. Some people confuse this cloning with genetically engineered animals, but it is not at all a genetically engineered animal. It is only an asexual means of reproduction where we have to provide the environment to a donor cell so that it can grow. Offspring of the clones are not clones. Aging has been found normal in clones. This has been proved by the group which produced Dolly as well as in many other species, and in India also there are 15-year-old buffalo clones. Clones and their offspring products are safe for human consumption. This was guidelines in the Food and Drug Administration of USA after conducting a series of experiments at different locations. Labeling is not required for clone animal products, and no indication that clones or their progeny would pose additional health risk or environmental risk. This has been also cleared by the EFSA. Many countries are using cloning technology to their animals. In the risk assessment study, which have been reported in cumulative in all these species, if we use the oocyte, it is similar like the IVF, so there is no risk. Donor cell, there is no risk. Cloning procedure also there is no risk. Surrogate animals, with our own experience and with the literature, we have found that the pregnancy rate is still low and there are comparatively more abortions. But we checked the number of abortions in the whole herd and compared, there was a difference but not that much. Health risks of clone animals is same as IVF animals. Progeny of the clones, there is no risk. So these are the animal cloning companies world over, and there are several more in India, because our research is under the government system, and we are doing in the government institution, particularly in the ICR institutions. So current focus of buffalo cloning research in India is the production of multiple clones, improvement in the efficiency, conservation, genetic, genomic and epigenetic process in clone animals, and health and performance assessment. Because the things develop with time. Buffalo is an animal which has 11 months gestation period, and it takes about 2.5 years to give 3 years to give a calf. So it takes its own time to study many things, but we are focusing on that. In particularly, we are stressing more on the bulls. And reason of cloning the bulls is that we are in shortage of quality bulls. And if you see, the semen requirement is increasing very fast. And now in India, complete AI has been approved by the central government, so there will be more requirement of the bulls and semen. So these are the available clones, males and females, their weight and details of the cells types which have been used and when these were born. I will simplify it and will show you the photographs of the clones available in our country. These are the bulls which are available at CIRB, Hisar. These are also available at CIRB, Hisar. These are the bulls available at NDRI, Karnal. These are also available. So, you can have a look that 7 clones from a single breeding bull were produced. This has been recorded in Limca Book of Records as well as India Book of Records of highest number of clones from a single animal. 3 clones from a single breeding bull, 4354. This 4354 bull is number 1 progeny tested bull of 15 set in the network project on buffalo improvement. So we are cloning the best animals. 2 clones 2501 reclone bull, and female calf also cloned from the top animal which was highest producer at CIRB since inception of the institute, and few more pregnancies are continuing. So these are the progeny of the clone. This is the Assamese clone which have been produced at CIRB. We have 4,000 doses from this bull, and there is one pregnancy also. And these are the multiple clones of M29 growing. These are the progeny which have been produced at CIRB using the cloned bull semen. And also we have an MOU with a dairy at Sirsa where they are having 500 animals. So lower picture is the clones progeny at high-tech dairy Sirsa. At NDRI also, dozens of clones progeny from the 2 bulls have been produced. Fertility of the clone bull. In 2019, we had a brainstorming session at Delhi. At that time, we had the fertility of only 1 bull. But now we have tested the fertility of all the clone bulls which are adult and producing semen. So both through IVF as well as through artificial insemination. And we could find that the fertility both IVF and AI based is equal to non-clone bulls. So this was published in Nature Scientific Report, a 5-year study. We could study the cell physiology, cell cycle, embryo development, epigenetic, gene expression, pregnancy rate, health of the clone bull, fertility of clone bull and calf, and we could conclude that growth, health and fertility of clone bull is similar to non-clone bulls. Clone bulls biochemistry, telomere length. This is very important. I will like to share it. Telomeres is related to the aging process, and in the adult animals there is different telomere length than the very young or embryonic growth at embryonic stage. When the donor cell is reprogrammed, the matured oocyte cytoplasm has
capability to reset the telomere length equal to the germ cells so that it can have the same age. So we have studied and compared the telomere length of the donor cells, embryos, and after 1 year, and we could find that it is matching with the age-related animals or the embryo. Similarly, global transcriptome analysis and GS targets that the expression of level of mRNA in spermatozoa of clone bulls and their somatic cells, donor bulls, was equal. Global mRNA and bulls. So progress till now, briefly I will summarize that 13 clones from 6 superior bulls, 7 clones from the M29, 3 clones from first rank 4354, and we are also working on a DBT project. I will show you 1 or 2 slides from that also. Now hundreds of progenies have been produced. We have 30,000 semen doses from the clone bulls. We have somatic cell bank. Male progenies of the clone bull are in semen production. Their semen has also been tested both in vivo and in vitro, and it is found equal. Female progenies have been also produced. Improved cloning efficiency from 1% to 6 to 8%, and fertility of the clone bulls is good. So, looking in all this, we submitted a proposal that how we can make more Indian data so that we can say that the clone bulls progeny and their success rate in AI as well as their performance is good or equal to non-clone bulls. So we could get some funds from the DBT, and we have done AI using this clone bull semen, and now we are in the process of studying. So total we have done 2,078 artificial inseminations from May 2002 to January 2003, and in different category of animals. And we could find that at overall 41.106% consumption rate was found in 996 animals which have been tested. So it is comparable because average, all India average of artificial insemination. So some calves have started coming because we started this project last year only. So now the calves are coming. So these are the trainings and demonstration. The program of animal cloning has been widely covered at all angles. Ministry has taken interest, Haryana government, central government has taken interest, and newspapers, in good newspapers it has been covered well. Also, current science, which is very popular Indian journal, they have published the SME buffalo clone on its cover page. And in Rajya Sabha TV, that has been also documented as a documentary. Now some people ask about the ethical questions. We try to study, read, satisfy ourselves, and then share our knowledge. So the main thing which comes is we need to build a societal consensus. Neglect of the issues has no ill effect on cloning science, but the sentiment could have effect on the commercialization and acceptance of the technology. So with this, I thank all of you for hearing. Thank you.
Any question from the audience?
Yes.
Very mathematics of the data. So I had regarding studies regarding the first thing. So having these studies regarding the, if we have superior bulls, is that thing getting transmitted? Do we have proof of the superiority in the breeding as well?
Till now, we have reports in progeny of the clone semen, male progeny, and we have tested all parameters of the semen, and all semen parameters and their IVF and fertility rates have been found comparable. Also, we have done some whole genome sequencing. For that, we have recently got some results. So we are in the process of analysis of that details we will be sharing, but it is comparable with buffalo genome.
So I fear that we cannot go for the wholesale use of this semen unless we are sure that this purity that we are claiming that is there.
Yeah, but unless until we will go ahead and produce and check in females also, we have few progeny which are giving more milk than their mothers. So rest the data. To answer your full question, we are generating more data.
Yes.
And this will be answered when more progeny which can have a significant data. And we can say that yes, this much number of progeny have been tested and found that they are giving better milk production.
So my point here is, today we know that genome is not the only thing that matters. The epigenome has its say as well. My question was in that context, actually. The epigenome, are we sure that the epigenome doesn't change as we are sure of the genome as well? Does the epigenome remain same in the cloned animals as the originators of them?
Some studies we are conducting on epigenetics also, and these will be in the process, but whatsoever we have studied, we have found it is okay. So there is also difference between the other animals also because of environment, progeny from the same animals or even in identical twins, there have been difference. Even in human beings, the identical twins, which we treat like clones, they have different fingerprints.
Yes, my point.
So these are the areas of study and these will be definitely taken care of.
Okay, sir. Thank you, sir. Wonderful presentation.
हाँ डॉक्टर साहब
सर,
हिंदी में पूछ लूँ?
आप पंजाबी, हिंदी और अंग्रेजी तीनों बोल सकते हैं।
नहीं आपको पंजाबी नहीं आती।
सवाल का जवाब तो दे दूँ।
एक बिल्कुल छोटा सा वो है।
हाँ जी।
सवेरे मदान साहब ने भैंस दिखाई थी और बछड़ा थी। अब आपने दिखाई है और वो दिखाई है। दोनों में नहीं कोई नहीं। दोनों में क्या अंतर है? या आपका कितना इम्प्रूव है या क्या है?
उन्होंने दिखाई थी फिजिबिलिटी, हमने दिखाया है कि मामला आगे बढ़ गया है। अब वहाँ रिव्यू क्या हुआ जहाँ था।
दिखाई पैदा तो उन्होंने भी की।
पैदा तो उन्होंने भी की थी, लेकिन अब उसका सीमन भी फ्रीज हो गया, उसकी प्रोजनी भी आ गई।
मेथोडोलॉजी वही है।
मेथोडोलॉजी भी।
May I have?
Please.
Last slide, you could see that you want that there should be a change in the policies to get your technology commercialized.
Yes.
Okay, that do you feel that technology which has a 6% success rate is going to be commercialized? When we heard a previous speaker that still so much advanced their technology and they are not going to have that commercialized. Do you feel that at 6% success story you will make this as a commercial?
It can be further improved, and the system in which we are working, you know that there is difficulty. Dedicatedly, the work is done on these aspects, there can be more improvement. Because as a scientist, I also have to do all those other things which are required in the system. So this is, if we dedicatedly, we focus on the second thing, answer to this question is, we can improve it, and at this efficiency also, if we can clone the best quality bulls, these will be very useful. Because this is not for all generations, but the best for 10, 15, 20 best broods of the country you want to clone, then it can be used and it can be beneficial also.
Thank Dr. Yadav.
Thank you, sir,
for your presentation. Now we have next speaker. Dr. Sandhya Chaudhary.
1 minute, sir. Somebody's car, MA's car, JK01AM3511. Is anybody is the owner of the car here? Thank you.
So that's why we work. यहाँ से क्या होगा ना नीचे पूछ के जाओगे बगल में एक
So, good afternoon, Chairman, Co-Chairman, Senior Professors, Dr. Mishra sir, Dr. J.B. sir, and my dear friends. Today I am going to speak on insulin resistance in dairy ruminants. Now, as we all know that the total milk production is increasing day by day in India. It has increased in 21-22 by 5.29%. Per capita has availability of the milk has also increased by 33.33%. The number of milk animals, they are also increasing. But what this data indicates that there is an increasing demand for milk production. And there is a need and urge for optimizing the dairy health of the animal. And the health of the animal is most important from the transition point of view. The transition period is the most stressful period during which the animal undergoes a lot of stress. Now this is just the overview of the glucose metabolism in case of the ruminants. The broader arrows, they indicate that how the gluconeogenesis from the different metabolites takes place and what are the metabolites that are utilized for the purpose of gluconeogenesis. As you know that the fermentation of the carbohydrate takes place in the rumen, there is production of the volatile fatty acids and mostly the propionate, valerate and isobutyrate which are used, the volatile fatty acids which enter into the blood circulation and from there it goes to the liver where through the process of gluconeogenesis it gets converted into the glucose. But there are other arrows from which also there are other things which can give rise to, or the non-carbohydrate sources which can give rise to the glucose. For example, the amino acids which are absorbed from the intestine, they can also give rise to the glucose through the process of gluconeogenesis. The glycerol is obtained from the hydrolysis of the fat from the adipose tissue that can be used in the process of gluconeogenesis. But once the glucose which has been available from the process of gluconeogenesis and during the transition period, this glucose is utilized to the two main organs of the body, that is to the udder as well as to the gravity uterus. So there are two things. One is regarding the glucose supply and another is the glucose removal. We say that the intake of the feed, it increases the glucose supply, but not only the intake but there are other substances, for example propionate, lactate, glycerol, amino acid, glycogen, all that can give rise to the glucose molecule. But the glucose which has to enter into the different cells of the body, it requires the protein which are the glucose transporters. And there are different types of the transporters. We usually classify these glucose transporters into two types. One which are dependent upon the insulin molecule for the transport into the cell and others which are insulin independent transporters. So the insulin, we are going to focus mostly on two tissues or the organs of the body. One is the liver, which is which can have the insulin independent glucose independent insulin independent glucose transporters, and the skeletal muscles and the adipose tissues which require the insulin dependent transporters. While this uterus, the udder, the placenta, they do not require the insulin dependent transporters, but they require the insulin independent transporters. So these are the different types of the transporters which are present. Now what happens during the transition period, I said that the transition period is the most crucial period because during this period there is physiological drift of the energy which takes place from the gestation towards the lactation. And we all know that the transition period comprises of 3 weeks before parturition to 3 weeks weeks after parturition. But what is critical is that a more amount of energy is required by the developing fetus. It is required by the mammary gland. It is required for the purpose of the milk biosynthesis. Further, it aggravates these conditions. It is aggravated because there is reduced feed intake. There is the stress of the parturition, and because of that, there may be losses due to the metabolic disturbances, which may be two types. One, which directly affects the milk in the immediate lactation cycle, and the second may be the indirect losses, which may be in the form of the certain diseases. It may be ketosis, it may be mastitis, it may be metritis, and which is going to affect the reproduction as well as the productive performance of the animal, and ultimately it may also lead to the culling of the animal. Now what happens, what are the physiological adjustments which are taking place during the transition period? Because the body has got its own system for partitioning of the energy, and this partitioning of the energy takes place towards the developing fetus as well as towards the lactating gland. And because this is accompanied by reducing the insulin mediated uptake in the body tissues, that is in the skeletal muscles and the adipose tissue, and making the glucose available to the non-insulin dependent cells of the body, that is towards the uterus as well as towards the milk production. So that over there arises the need for the insulin resistance. So before discussing the insulin resistance, what are the major functions of the insulin? We are going to talk about the insulin dependent cells and insulin non-independent. If we consider in case of the liver, what it does, it causes the stimulation of the glycogenesis, means the synthesis of the glucose. It helps in the synthesis of the triacylglycerols. It helps in the synthesis of the proteins. It helps in the process of glycolysis. But it suppresses what? It suppresses the gluconeogenesis. It suppresses the glycogenolysis, and it suppresses the ketogenesis as well as the degradation of the proteins. In the skeletal muscles, it is not going to help in the synthesis of the triglycerides, but it is going to help in the utilization of the ketone bodies, and it will help in the stimulation of the uptake of the glucose because it is insulin dependent cells. There will be separation of the protein degradation and glycogenolysis. But in case of the adipose tissue, there will be only stimulation of the uptake of the glucose, lipogenesis, and the lipolysis will be suppressed. Now, what is insulin resistance? Insulin resistance means whether a normal concentration of the insulin it does not able to produce the normal biological response, or it is capable of producing a reduced biological response. Now you can see the two diagrams over here. One over here you can see is the insulin sensitive, another is the insulin resistance. Insulin sensitive means when a small amount of insulin is required to push the large amount of glucose into the body, while in the second one, in case of the insulin resistance, require large amount of insulin to push the same amount of glucose into the body. Now this can becomes more clear with the help of this graph. You see that on the x-axis we find the hormone concentration, on the y-axis we find the biological response. On the x-axis, as we increase the hormone concentration, or we say that a normal concentration of the hormone elicits 100% biological response. While in case of insulin resistance, there are two terms. One is reduced insulin sensitivity, and what is this reduced insulin sensitivity? Means more concentration of the hormone is required to produce that 100% biological response. That is what is the reduced insulin sensitivity. The second thing is the reduced insulin responsiveness. Now this reduced insulin responsiveness means less amount of insulin is produced, so less is produced, so less amount of glucose can be transferred. And in the third condition, there may be both decreased sensitivity as well as the reduced responsiveness. So why this need for the glucose diversion arises. We have already seen that the insulin resistance is necessary because during this the preferential diversion of the energy takes place. But during this time we have seen that the number of the insulin independent transporters, it increases 3 times. And second thing is that during this there is inflammation also increases, and the inflammation is responsible for causing the production of the pro-inflammatory cytokines such as tumor necrosis factor alpha. Then there is a controversy about the immune suppression as well as the diversion of the energy towards the immune system. Bumgard, he has worked a lot on the oxidative stress, and Bumgard, he says that when there is insulin resistance which is taking place inside the body, the transfer of all of the nutrients the glucose, it gets transferred towards the immune cells, and because of that there is no transfer of the glucose towards the milk production, and because of that there is decrease in the milk production. And hence immune system is compromised. Sorry sir, I am really having a sore throat, so it is becoming a bit difficult for me to speak. थोड़ा पानी दो सर, cold water. Cold, I don't want cold water. Now this endocrine regulation is important for insulin resistance, but we say that insulin resistance is a homeostatic mechanism. But what is homeostasis? Homeostasis is a orchestrated or a coordinated event of the metabolism which is going on inside the body to support the physiological state of the body. There are 3 things which are responsible. One is insulin, second is the growth hormone, and third is the responsiveness of the body tissue. Now, pituitary gland is the main endocrine gland which controls the process of galactopoiesis as well as lipogenesis, and that is through the growth hormone. And this growth hormone now this growth hormone, what it actually does, it stimulates the production of the insulin-like growth factor 1, and this insulin-like growth factor 1 again has got a negative feedback mechanism on the growth hormone. So growth hormone has got a diabetogenic effect. It produces its effect antagonistic effect to that of the insulin hormone. So what it does, we say that insulin is responsible for lipolysis, growth hormone is responsible for causing the lipolysis. It decreases the lipogenesis also, and what it does, it reduces the transport of the glucose transporters to the adipose tissue, and because of that there is reduction in the glucose uptake by the adipose tissue as well as the skeletal muscles. We have seen that the growth hormone, high amount of the growth hormone is necessary for the milk production. But what happens? There is uncoupling of the somatotrophic axis which takes place at the time of parturition. Suppose we say there is a normal level of the growth hormone which is present over there. But this growth hormone receptor 1A, there is down regulation of this receptor. Because of the down regulation of this receptor, there is decrease in the insulin like growth factor 1, which removes the negative effect, and because of that, there is increase in the growth hormone which takes place at the time of the parturition and which is responsible for causing the insulin resistance. Now, we say that insulin resistance is usually seen in case of high yielders. Why? Why it is seen in case of the high yielders? It takes place usually in case of the high yielders because what happens? The glucose is diverted towards what? It is diverted towards the memory gland. It is diverted towards the uterus. So what happens? Animal feels that there is low blood glucose. And when it feels that there is low blood glucose, it will simulate the situation similar to that of the lower feeding. That is why. So the normal level of the blood glucose, it acts as a stimulus, acting just like the lower feed intake. And this is the reason why high milk yielders, they are having more insulin resistance as compared to the low milk yielders. Now see, sir, we have got a lactation cycle. In this lactation cycle, we consider 3 periods. One is the fresh period, immediately after parturition, 2 to 3 weeks postpartum. There is increase in insulin and growth factor 1. When this increases, what it does, it increases the number of the memory epithelial cells. It increases the secretory activity of the memory gland. Now during this period, although the insulin concentration is low, but though the insulin concentration is low, but the liver size is going to increase because the gluconeogenic capacity, it is going to increase. So insulin remains low, insulin resistance is high, insulin like growth factor is low, growth hormone is high, GLUT4 expression decreases, that is towards the skeletal muscles and the adipose tissue, but GLUT1 expression in the memory gland increases. So what is the net result? The net result is that there is increase in the glucose production, mobilization of the body reserve takes place, and the energy is used for milk production rather than towards the muscles. But in the early lactation also, the energy is used for the milk production, but then also the sensitivity of the insulin starts increasing and the insulin concentration also increases, although the partitioning of the energy still just now is towards the milk production rather than towards the body reserves. But when we go towards the mid or the late lactation, partition of the energy goes towards the body reserves because the insulin concentration now starts increasing, the insulin sensitivity decreases, and the GLUT4 expression of the transporters increases. Sir, I wanted to speak a lot, but I think the time does not permit. So I will just try to finish it off as early as possible. Only 5 minutes I will take, sir. Because this NEFA, what is the role of NEFA? Because we say that nowadays testing of NEFA as well as BHBA is going on the commercial farms for knowing the subclinical type of the ketosis. So we say that the NEFA concentration increases and the negative energy, NEFA concentration increase indicates the negative energy balance. But how much should be the NEFA concentration increase? We say that there is a cutoff limit of about 700 micromole, which is considered as the cutoff limit for the negative energy balance, and 1.2 millimole per liter, that is considered as a cutoff limit for the BHBA. But many times it has been seen that the high producing animals, they are having high concentration of NEFA, but still the milk production does not decrease. Another case that they are having high concentration of BHBA, but still again the milk production does not decrease. So many times the farmers, they raise the question that in foreign countries they are raising this type of question that my animals, they are showing high BHBA, my animals, but still my animals are milking crazy. So that means the animal is capable of handling the negative energy body balance, so that it does not mean that we should not go for the testing of the BHBA or we should not go for the testing of the NEFA. It should be, it is usually done for the epidemiological studies. So it would be of concern if the concentration goes on increasing for a longer period of time, but if it decreases, for example, in this graph, you can see that the concentration of the NEFA increases on the day of parturition, but then it starts decreasing. If it starts decreasing, that means the animal is going towards the positive energy balance. So because the NEFA has got the three main role, it can be utilized for oxidation, it can be utilized for the synthesis of the triacyl glycerols, or it can be utilized for the production of the BHB. So they are the three main things where the NEFA gets used. And if you see that during this, on the day of parturition, that is about 15 times increase in the uptake of the NEFA by the liver. So when this takes place, it can result into the deposition of fat inside the body. So sir, I will just conclude it. So what is important is that not NEFA is not much important. What is important is the hepatic energy charge. Means if more amount of energy, if more amount of ATP is available, then what will happen? Even if more amount of acetyl-CoA is there, then the less amount of feed intake will take place. But if less amount of hepatic energy charge is there, then more feed intake is going to take place. And so there are different types of indexes which are being used for testing the insulin resistance, which are based on the calculated values. For the gold standards, they are the HEC, that is the hyperglycemic euglycemic clamp, hyperinsulinemic euglycemic clamp, glucose tolerance test, which is being used. And but these indices which I want to say, these are the indices which are being used. That is Quickie, HOMA-IR, Quickie NEFA, and Quickie BHP. HOMA-IR has got a direct relationship. Over here we take into consideration the glucose and the insulin. But in case of Quickie, we take the log concentration. And there are revised, then the Quickie has again been revised, and that is why it is known as R-Quickie, where we have taken into consideration for NEFA as well as and R-Quickie is BHBA, which is for the beta hydroxybutyrate. So, but whatever the studies, they indicate that R-Quickie BHBA is a better indicator, and it has got a good correlation with the hyperinsulinic equality. So what is important is ameliorative strategies should be used. That is how we can prevent the negative energy balance and increase the insulin secretion of the body, and there should be an alternate energy source for the negative energy balance. Now actually what important thing when scientists say is that whatever the NEFA goes to the liver, this in the liver this NEFA can be converted into fat. But what is important is the removal of the fat from the liver, and that can be done with the help of the using certain such substances such as choline, which are lipotropic in nature. So, sir, I will just complete it. Thank you. Because I required more time, it will take more time. Okay, thank you, sir.
So, any question from the audience?
And you have studied the insulin concentration? If so, at what time you have studied after feeding?
Sir, we are doing it early in the morning. We are feeding the animals, and early in the morning we have taken the insulin. We have taken the blood samples.
Because the insulin concentration will be fluctuated depending upon the status of the animal.
Yes.
Feeding status of the animal.
Yes, sir.
On the other hand, if the insulin is going to affect on the glucose concentration,
Yes.
then it will be affected on the NEFA itself. Because
Yes.
there will be the mobilization of the fat
Yes, sir.
for the energy need.
Yes, sir. So that is why we have taken in the early morning, without feeding the animal.
So there will
Means in the evening the animals, they are not fed, and then up till in the morning they are not.
correlation between insulin and the NEFA.
Sir, insulin concentration, actually when the insulin concentration decreases, we say that NEFA increases. This is what.
What is the normal concentration of blood glucose, madam?
Sir, it varies from species to species. Suppose if you want to consider the ruminants.
No, ruminants I am asking.
Yes, sir, it is about 40 to 70 milligrams per 100 per deciliter.
Because they are less dependent on insulin.
They are dependent on the process of gluconeogenesis.
Not much like human beings are monogastric.
Yes, yes. That is why, because propionate, it gets converted as a gluconogenic fatty acid, it gets converted into glucose. So that is why we are having 3 fatty acids are produced. But out of that, only one is glucogenic, and therefore the concentration of glucose in case of the ruminants is less.
मैं कैसे ले सकता हूँ सर जब बोलता हूँ
Yes.
Since you have discussed physiological states,
Yes.
pathological states associated with this kind of situation, like diabetes.
Oh yes, of course, pathology is completely different. But when we are talking about the physiological state, we consider that lactation, then we consider gestation. These are the physiological states.
Are those conditions similar to those of humans?
No, it cannot be similar to that of
I
human.
mean, in broad etiology and in broad treatment possibilities.
In case of, when we are talking about the insulin resistance, in case of type 2 diabetes, this insulin resistance is studied in case of the human beings. And over there it is due to the obesity which is taking place. And that is responsible for causing the insulin resistance in case of the human being.
So do we have such situations in animals?
Yes, we are talking about the BCS. We are talking about the BCS.
Okay, thank you.
Okay.
Thank you.
Thanks, ma'am. Sanjay Dalvi. Now, we have come to the end of the session. After 3 presentations were there, all were presented. And with this, the topics were different. One was on biosensors application in the detection of pollutants and other, pollutants and adulterants in the milk and other things. Second was on the cloning by Dr. P.S. Yadav, and third was by Sandhya Chaudhary on insulin resistance. So all the topics were covered nicely, and we thank the presenters for presenting their papers on as lead papers. Thank you very much. So with this, we close the session.
So before we conduct the parallel sessions of oral presentation and poster presentation at the outside, so Faculty of Horticulture, yes, I would request Chairman of the committee, Professor S.K. Rastogi sir, to felicitate our speaker, Dr. P.S. Yadav. Yes, yeah. I would like to invite co-chairman of the committee this session to felicitate Dr. Sandhya Chaudhary. I would like organizing secretary to felicitate the chairman of this session, Dr. Professor S.K. Rastogi, sir. And after that, to the co-chairman also. So ladies and gentlemen, we will take a break here for 5 minutes so that we will make the arrangements to conduct the sessions here. One another is the VC secretary, and third one is the horticulture building. Their poster sessions will be held. So thank you so much.